there's two marks here. This is the distal balloon. This is the proximal balloon. The way that these work is the distal balloon ends up blowing up distal to this marker. The proximal balloon dilates proximal to this marker. So it's kind of opposite. So again, so when we blow these up, you'll see that. But the balloon, there's going to be one balloon here, and we're going to infuse chemo between the two balloons here. And what we're going to do now is we're going to pull back this Renovo catheter. You can see here. Now, the next thing we're going to have to do is we're going to have to pull back the sheath a little bit. So first thing I'm going to do is I'm going to try to blow up this distal balloon here. And you can watch, maybe I can mag up one more to make it a little bit more apparent here. Okay, and then I'm going to pull it back even more. Now, another technical tip here is, again, remember the proximal balloon is going to be proximal to this marker. Again, and right now if I blow up the proximal balloon, the sheath is going to cause it to rupture. So that's another important technical thing. So if I just blow it up now, the balloon will break for sure. So when we blow up the proximal balloon, we're going to pull back on the sheath a little bit. What I'm going to do right now is first we're going to blow up the distal balloon here. So I'm going to show you real time here. And again, it requires very little volume. Remember, maximum is really 1 mL. You want to go about 0.2 at a time. You can see the balloon going up there. A little back tension on the... You can see me blowing it up here. I've done about 0.5 mLs here. And the maximum volume that you could put in here is about 1 mL. And you could still see kind of things moving, still not stagnant. I kind of want it to form a little bit of a wedge. There we go. You kind of see it forming along the artery itself. And I'm going to leave that there. You can see kind of moving around less. I'm sure there's still going to be a little flow around it. We're going to have to blow up the proximal balloon. So now this is the proximal balloon here. So we're going to pull back that sheath. So you want to go ahead and pull it back. Pull it back. Okay, hold it right there. I'm blowing up the proximal balloon there. Back tension on the whole catheter. Okay, there we are. Pull back on the sheath a little bit more. Okay, leave it right there. Let's see how this goes in here. Back tension on the catheter, please, on the Renovo catheter. Okay, you can see the two balloons there. I'm going to take a single shot there. And now we want to see, is there flow between the two? So we're going to inject in the chemo infusion port, which is the red one. And I want to see a stagnant column of contrast. I don't think we're going to achieve that yet, but let's see what it looks like. I'm injecting. And I don't see a stagnant column. And I don't know if it's washing out distally or not. We could do a little run here. I want you to hold your breath, my friend. Don't breathe, don't breathe, don't breathe. I'm going to do a little run here. You could see contrast washing down. See that? So this is not where we want to be yet. We need to blow up this distal balloon a little bit more. So that's what we're going to do here. Apply back tension. Back tension. Need a little more volume. Okay, we'll put a little more volume in there. Now I'm going to inject contrast again between the two. The other thing I could show you that we could do is we could also, if we want to see if the proximal balloon is occlusive, is I can do a run through the sheath. So hold your breath. This is a run through the sheath. Hold it, hold it, hold it. So you can see a proximal balloon is occlusive, right? We're not seeing any contrast going through there. Now I'm gonna, having blown up this balloon again, keep that back tension. I'm injecting now between the two balloons again through my chemo infusion port. And look at that. We have a stagnant column of contrast. This is where we want to be. This is perfect. Now we're ready to infuse. Chemo, please. We're going to maintain that tension at all times. And as we infuse the chemo is not radiopaque, so it's going to wash out. The point is we want to kind of know what it looks like and maintain the tension. We want to make sure the balloons are not rupturing as we connect this. So we're going to do this. Okay. Keep going forward. Okay. And now let's arm it. Can we start it, please? You're good. We could go ahead and start it. Yeah. You got notice. Yes, go ahead and start, please. Notice, look, we still have that stagnant column of contrast. This is exactly what we want. This is perfect. Now, we're giving the chemo here, and as we give this, and we'll take some images along the way, that contrast is gonna wash out because there's no contrast in the chemo. So the only way we're gonna just keep an eye on it. Dr. Overfield is really kind of holding this in place, and we're gonna just intermittently fluor to make sure the balloons haven't popped, nothing has moved. But this is, you can see the contrast has now washed away between the two balloons, and that's what we expect because the chemo is going in. And we're just going to do intermittent images here over the next 20 minutes as the chemo is being infused. And the other thing I want you to notice is the patient tolerates this very, very well. We're just doing this with moderate sedation. You can see he's following all our instructions and so forth. And now it's just a waiting game. We're giving the chemo over the next 20 minutes. But again, the key in this procedure is achieving that stasis of contrast between the two balloons to make sure there's no branches that are in between, which we initially did not achieve. The other thing you kind of want to look at is just kind of troubleshooting, and from having done a decent number of cases that we had one case where we had some chemo leaking. So just really basic things, but just making sure that this is tight here. At where the chemo is connected to the actual catheter itself, or the tubing, I should say. And then you come around here. In this case, we have an extension tubing, so you want to make sure it's not leaking at the connection. And the only other place it could potentially leak is right at where here we have it connected to a 3-way stopcock and to the Renovo catheter at the red line.